to have a better understanding of ecosystem uptake and provide insights into different types of model formulations. So with that short background, I'll just give a brief overview of how we plan to organize this session. We have 9 excellent speakers lined up for today's oral session, and each speaker will have around 9 minutes, at which point my co-chair, Nick, will actually give a verbal warning at the end of the 9 minutes, and then we'll have a quick 3 minutes for question and answer before moving on to the next session. We'll do 4 talks and then we'll take a quick break. I understand it's always really long to kind of be sitting in the computer for 2 hours at a stretch. So we'll do 4 talks, take like a 5-minute break, and then we'll come back for the remaining 5 talks. So without any kind of further ado, let's just get on with the first presentation. And if you have any questions for the speakers, then please make sure to put it in the chat if it's immediate, and then we'll try to take a look at it once we get to the end of that presentation. So with that, now our first speaker will be Anna Garaskina, and from Center for Forest Ecology and Productivity in Moscow, and she'll be talking about forest types and soil carbon stocks in European Russia. So over to you, Anna.
Okay, thank you very much. Forest types and soil carbon stock in European Russia. I'm sorry. Next. Research relevance. Boreal forest soils are a significant contributor to global carbon storage. Soil and vegetation are mutually associated with each other, but linking forest types and soil carbon stock is not clear. There is evidence of the effects of tree species on carbon stock in organic horizons, but is little known about this effect in mineral horizons and about the effect of ground vegetation on soil carbon stock. And it is important to understand how soil biota regulates these effects. And our research aim is identifying links between forest types and soil carbon stock in European Russia. Our object, we conducted our research in taiga forests. There are Leningrad region, Republic of Karelia, and Karelian Isthmus. And we studied conference broadleaf forests. There are Moscow region, Bryansk region, and Northwest Caucasus. In taiga forest, we studied our research on the regular grade, and in this area, dominant trees were spruce, pine, birch, and aspen. The type of soils are albic podzols, rentic podzols, and albic retinols. There are widely distributed in this area. Conference broadleaf plain forests we studied in Bryansk Polessia. It is a protected area. We conducted pine forest, pine broadleaf forest, and broadleaf spruce forests on albicals according to IBFRA2021. And in Moscow Oka Plain, we studied bush lime forests and spruce broadleaf forests on albic retinols. Conference broadleaf mountain forest we studied on Northwest Caucasus. The main type of forest in this area are hornbeam aspen forests, hornbeam fire beech forests, and beech fire forests on district Cambizols. We used geobotanical description in which type of communities to provide vegetation study. We studied soil macrofauna, especially soil macrosaprophages, such as woodlices, millipedes, mollusks, and earthworms. And soil study. Samples of soil organic horizon we divided by two layers, L layer or little layer and FH layer, and mineral horizons A, AE, E, and B. The first results are about soil carbon stock in taiga forest. The main results on the first main results on this area is the soil carbon stock was associated with soil types. The carbon stock in carbic antic podzols was the highest compared to carbic albic podzols and albic retizols. We can see the differences in results. The second main result is the carbon stock was significantly higher in spruce forest than in pine forests on the same soil types. And the third main results in taiga forests is carbon stock was significantly higher in forests with more proportion of herbs in drought vegetation compared of green mosses forests. In broadleaf coniferous forest, we had some differences in soil carbon stock in organic horizons and mineral horizons. In organic horizons, differences are connected with type of forests, region, type of soils, and activity of soil biota. About activity of soil biota will be in next slides. The main results about mineral layers is the highest soil carbon stock and mineral layers were found in the mixed forests producing litter 4 of different quality. Quickly and slowly decomposition litter. For example, in Northwest Caucasus, there are Horbian fire beech forests with different litter quality. In Bryansk Polessia, pin broadleaf forests, and on Moscow Oka Plain, spruce broadleaf forests. Next, we examined predictors to provide soil carbon stock in soil organic horizons and mineral horizons. And we got that the most important predictors to soil carbon stock in organic horizons are base saturation in little layer, CN ratio in little layer, and biomass of functional group forms associated with the organic horizons. We choose only worms because this group was predominant in biomass among another groups. Biomass of forms was about 90–95% of total biomass soil macrofauna. And the most important predictors to soil carbon stock in the mineral layers were base saturation in little layer, species riches of woody plant in herb layer, and biomass of functional group of worms associated with soil mineral horizons. And linking between functional group of worms and carbon stock in organic horizons. We can see the carbon stock in organic horizon closely and negatively related to the biomass of functional group of worms that process the litter. There are epigeic, epientogeic, and anegeic worms. In mineral layers, the carbon stock related to the biomass positively of functional group of worms whose life activity related the mineral part of the soil. It is endogeic group because this group is humus forming group. And the main results and conclusion, the study carried out in the dominant forest types of the European part of Russia confirmed the influence of climate and soil forming rocks of the soil carbon stock and demonstrates the links between vegetation, earthworms, and soil carbon stock. The soil carbon stock are regulated by the litter quality of trees, growth vegetation functional group, and activity of soil biota. The important prediction of the soil carbon stock variation are the litter quality indicators such as base saturation and CN ratio. In taiga and conference on broadleaf forest, the highest carbon stock in the organic horizon was associated with the low quality litter. Among taiga forests, the highest carbon stock were found in spruce forest with higher proportion of herbs and growth vegetation. Among conference broadleaf forests, the highest soil carbon stock in the mineral layers were found in the mixed forest producing litter flow of different quality. In conference broadleaf forests, the carbon stock in the organic horizon were closely and negatively related to the biomass of one group of worms that process the litter, while the carbon stock in mineral layers was closely and positively related to the biomass of functional group of worms whose new activity is related to the mineral part of the soil. It is an again group. The mixed litter flow consisting of the fast and slowly decomposed components produced by plants belonging to diverse woody and herb species. It is a prerequisite for active functional earthworms related to both organic and mineral soil layers and for high soil carbon stock. Because it is only mixed litter provide optimal trophic and tropical condition for earthworms. Thank you very much for your attention.
Thank you, Anna. Thank you for that great presentation. So there are a couple of questions that have come through the chat. So let me ask them here. And then if anyone else has questions, please put them in the chat or feel free to raise your hand so that we can identify who's asking the question. So the first question comes from Georgie Alexandrov asking, did you take into account the position of plots in the landscape that is on Catena?
Okay.
Yes, may I shortly to explain this question. Perhaps you remember the name Natalia Basilevich, who was many yes, and Natalia Basilevich was always said that it's if you consider soils, you should always pay attention to the position of your plots within the landscape. Is it on the upper part of the landscape? Is it on the middle part of the slope or in the end? Because especially in Karelia, that was the works of Monakov. Of course, it's also world study and probably you did not know about them, but they show very close correlation between the position of the plot on the slope and the accumulation of carbon in the soil's horizons. So, and so that's, of course, maybe in your classification, you use, yeah, there is some correlation between the forest type and the position on the slope, but probably, but did you consider it explicitly in your analysis of the data, because it's a very important factor, especially in case of Karelia?
Thank you very much for your questions. Yes, it is very important, the position of plots in the landscape. I don't know exactly the answer of these questions. Unfortunately, I didn't work in this area. I worked in confidence broadleaf forest in the Bryansk, Polessia, Moscow region, and Northwest Caucasus. With I don't know exactly.
I'm just wondering, like, could you provide like a quick summary of that answer, since we are right on time? So I would like to move on to the next speaker. So if you could please respond in the chat so that everyone can see the answer. And then there is also a second question from Jill Johnston that if you could again take a look at in the chat and then respond, then that will help keep the conversation going.
Okay.
And I'll take control of the screen. Or sorry, can you stop sharing your screen so that I can move to the next presentation?
Okay.
So it's on top of your screen. Normally on top of your Zoom meeting screen, you should have a stop sharing red rectangle.
Okay.
Okay, thank you, Anna.
Okay, great. Thank you, Anna. And our next speaker is going to be Anastasia Maknaikina from Siberian Federal University. Anastasia, do you want to share your screen?
Yes, sure. Hello, everyone.
Hi, Anastasia. Yeah, it would be great if you could share your screen, because for some reason Zoom isn't allowing me to share my screen, which I don't know what the problem is. So.
Okay, do you see it? Yes,
yes,
no?
Yes,
I can.
Yes.
Yes, perfect.
Okay,
thank you.
Great. So I'll stop my video during the presentation, then I'll turn back. So, hello, dear colleagues, one more time. And today I would like to underline another part of carbon in the forest ecosystem, which is soil respiration or CO2 emission. And during my talk, I would like to concentrate on respiration, water interaction during the vegetation season in the lichen pine forest of central Siberia. So as you know, soil emission, it's a very complex process, which is consists of many processes such as food respiration, mycorrhiza, and the plant activity in the soil, and the other heterotrophs activity in the soil. But here we should also remember about some factors, some external factors, which are human activity, such as logging, fires, pollution, and another type, natural factors, temperature, precipitation, substrate, or elements of mineral nutrition. And today we will try to understand how precipitation will change the soil emission in the forest ecosystem. So the aim of our study was to find out the response of soil emission to the rain events in the lichen pine forest of central Siberia during the vegetation season. In terms of this question, we concentrated in the following tasks: observe the seasonal and diurnal soil emission in the lichen pine forest, estimate the impact of rains to the emission rate during the season, during the summer season, and the next so-called birch effect. So the birch effect showed how moisture regulates the soil emission. Our study area was actually like a graphical center of our country. It's middle taiga forest, and we did our research and the station which called POTPO. It's an international project with the Max Planck Institute, Germany. So the study site was located in the lichen pine forest. It's actually one of the main forest types 190 years. Observation, as I said, during the summer season up to the end of September, and the frequency seasonal measurement was once a week, and diurnal every day, 24 hours. So research methods, we used direct measurements. So for it, we used Flycore gas analyzer, and we used types of cameras: dark cameras, this one, and light cameras. Sketch also some photosynthetic activity. And also we measured soil temperature during the season, and volumetric soil moisture. Actually, we have data from 2012, and here is presented for 2015. But today, I just wanted to talk about the last season, 2020, which actually was different from the mean values. The precipitation was around 20% higher, and the temperature too during the season, during the summer. So, and let's go to our results. So first of all, I'd like to present to you the seasonal emission during the summer. And actually, it's a kind of normal dynamic because the peak of emission we observed in the middle of the season in July, in the second half of July. And the difference between the fluxes around 8-10 micromole. It's quite a lot, I would say. Later, we look at the dependence between the soil emission and some meteorological parameters, soil temperature and soil moisture. We found some small relationship with the temperature, some exponential. And with the moisture, we found parabolic dependence with moisture with some optimal parameters around 30% of the moisture. Later, look at some interesting event. So we measured the soil emission after big rain event. What is the big rain? We call it heavy rain. So precipitation should be more than 10 millimeters per one time. And after this heavy rain, we observed enhanced or increase of CO2 emission up to 12 times. First 3 days after each this big rain event. So as you see, it's really a lot of CO2 which released after this big rains, which we can detect during the seasonal measurement, and even more from the model observation too. Just from the direct measurements, daily measurements, we can catch this CO2 releases. yes, during the first 3 days. So later we estimated how big this emission during the season for each month, and we found the biggest input to the month's emission observed in September, around 60%, and during the season, the mean impact from the rain flux, we so called it, around 45%, which is really a lot, which is, yeah. And here I'd like to sum up my short talk. So seasonal course of emission with a peak of fluxes in the middle of the season. During a variation of emission effect of heavy rains, which we observed, so heavy rain lead to rapid impulse response of CO2 emission, and as a consequence, the release of additional CO2 from the soil into the atmosphere. And so, of course, daily resolution can provide this information about sharp CO2 emission releases. Thank you for your attention.
Great. Thank you so much, Anastasia, for that wonderful talk. Are there any questions for Anastasia? We have a couple of minutes.
Hi, Anastasia, this is Scott Getz. I have a quick question for you, just a clarification. If you could back up a couple of slides to your results where you show the effective rains on CO2 emissions just thereafter.
Probably.
Yeah, this one, yeah. Can you go to slide? Yeah, so I just happened to see that you had sort of the biggest precipitation event didn't actually appear to produce a CO2 emission spike like many of the others. Do you have any thoughts on that one? Is it just a data glitch, or is it just that's the way it was?
I'm sorry.
You have
yeah, I have some problem with the microphone. Maybe could you shortly repeat or just write it in the chat? I'm sorry.
Okay, I'll try once more while other people put their questions in the chat. You have a precipitation event of like 32, like the highest one.
Yeah, the highest one, yeah.
But there's no CO2.
Yeah, okay. Now I see.
Just curious about that particular situation. Any thoughts?
Probably, as you see, because it's a small scale, you see here, we have some gap in measurements. That's why there was some problem with our device with the LICOR. That's why we didn't catch it. Probably there should be some emission. That's
okay,
why.
I thought it might be something like that. Thank you. Nice presentation.
Thank you.
Yeah, thank you, Anastasia. And there are a couple more questions for you in the chat. If you're able to see them, please take a quick moment to look at them while we move on to the next presentation. And if you could kindly respond to those questions in the chat, then that would be great.
Yeah, sure.
Thank you, Anastasia. And if you could stop sharing the screen, then we can go to the next presentation.
Yeah, okay.
And our next presentation will be by Ali O'Neill from Michigan Technological University.
All right, hi, can everyone hear me all right?
Yes, we can hear you. And you are full screen and good to go.
Okay, great. Let me just turn off my video so that I'm not blocking my screen. Okay, thanks. All right, so yeah, I'm Ali O'Neill. I'm a third-year undergrad at Michigan Technological University, and I'll be talking about some research I did with the U.S. Forest Service on whether spectral indices can detect changing CO2 and CH4 cycling in response to hydrological stress in northern peatlands. So as some of you probably know, peatland ecosystems play a huge role in the global carbon cycle, with carbon stocks rivaling those of all of Earth's forests, despite taking up only about 3% of global land area. However, this carbon sink status is vulnerable as climate warming, altered rainfall patterns, and drainage for agriculture and forestry all threaten to lower water tables. Although we've learned that remote sensing might be the perfect tool to track changing carbon cycles in peatlands, which are often vast, remote areas that are hard to access. And we already know that hyperspectral indices can successfully detect changes in surface water content and water table depths. And as it turns out, this unassuming little sphagnum moss may be the perfect remote sensing target, not only because it's often referred to as an ecological engineer for its role in the formation of peatlands, but because it lacks a vascular system, so it relies solely on capillary action for the movement of water, making it very vulnerable to predicted water stress and creating an easy target for hyperspectral indices to detect. And another cool thing about sphagnum is that different species grow in different landform types. So they may react to water stress differently, possibly causing carbon cycles to be affected differently. And so this is what I mean by different landform types. On one end, on the left, you have hummocks, which tend to grow higher above the water table and allocate more resources to growth in height. So their capitula, or that cluster at the top of the moss that's largely responsible for photosynthesis, tends to be smaller and more densely packed, which may improve the efficiency of capillary action. While on the other end, there are hollows or lawns, though I'll be referring to them as hollows, that grow much closer to the water table and tend to allocate more resources into larger, less dense capitula. So in this study, we wanted to know how the carbon dioxide and methane cycling of the two contrasting sphagnum landform types would be affected by water stress inflicted by lowered water tables and drought. And we also wanted to know if remote sensing techniques could not only be used to detect surface moisture levels and water table depths, but if they could be used to detect and track changes in carbon cycling during times of water stress. So there were actually two mesocosm experiments included in the study, but I'm just focusing on one for the sake of time. We sampled 32 peat monoliths, which we called sphagnicosms, from both hummocks and hollows from a sphagnum-dominated peatland in Astoria, Michigan in the United States. And we removed all vegetation except for sphagnum. And then we imposed a high and low water table treatment and an average and droughted rainfall treatment. And I did want to acknowledge that there's some uncertainty regarding future rainfall patterns. So we chose our droughted treatment solely to inflict water stress. And then our average rainfall treatment was based on average summer rainfall in the study area. So we conducted CO2 and CH4 flux measurements using a Picaro gas scouter in both the light and dark, using a blackout cloth so that we could measure both photosynthesis and respiration. And then we also took hyperspectral measurements using a handheld spectroradiometer. We used two indicators of vegetation greenness, chlorophyll index and normalized difference vegetation index, and then two indicators of wetness, so wetness index and floating water band index. So this figure is looking at net ecosystem exchange, which is CO2 minus energy loss from respiration. So the green boxes represent the hollows that grow closer to the water table, and then the brown represent the hummocks. So the high water tables are marked with an H and the lower marked with an L. And then as you can see, each set of boxes is sectioned into either average or droughted rainfall treatments. So any NEE below 0 represents a net uptake of CO2 into the ecosystem via photosynthesis, while any positive NEE represents a net loss of carbon dioxide to the atmosphere via respiration. So it's clear that low water tables have a substantial impact on hollows, as both low water table treatments had a positive NEE regardless of rainfall treatment. And then you can also see that hummocks had a negative NEE regardless of treatment. And it isn't really until both the low water table and the droughted rainfall are combined that the hummock NEE begins to shift to positive. So this shows that hummock CO2 fluxes are much more stable under water stress, indicating that they're probably just more resistant to water stress in general. And then here we have a figure showing methane flux data, very similar in setup to the last figure, except this one is showing methane emissions. So you can see that methane emissions were highest in the high water table average rainfall treatments and in all the high water table treatments, which makes sense when we think about methanogenesis, which occurs in the anoxic zone. And then overall, you can just see that methane fluxes were higher in the hollows than in the hummocks. And then this figure is showing us that spectral indices were able to accurately detect water stress in our samples. The top of the figure is focusing on hummocks, while the bottom is focusing on hollows. And then picture A and C show samples that received low water table droughted rainfall treatments, while B and D received high water table average rainfall treatments. And you can see that there's a stark difference between the hollow samples, but it's much harder to tell the difference between treatments in the hummocks. And the spectral data is telling us the same thing. So this figure is average from all of our samples and spectral measurements. On the y-axis, we have reflectance on a scale of 0 to 1, with 1 being 100% reflective. And then on the x-axis is wavelength. So the green bands show the range of the vegetation greenness indices, and the blue show the range of the wetness indices that we were using. You can see in both bands, there's a little dip in the figure. And so those are the greenness and wetness dips. So the steeper the dip, the more green or wet the moss is. And you can see in the bottom half of the figure, that orange dashed line representing hollows with low water table droughted rainfall treatments. There's hardly any dip in it, telling us that hollows with that treatment were very dry and bleached, just like you see in picture C. And then I also wanted to point out how far apart each treatment level in the hollows is. Well, there's much less of a difference in indices between the hummock treatments, indicating once again that hummocks are more resistant to water stress. This figure is comparing spectral indices by treatment, so sphagnum type, water table, and rainfall treatment. And as you can see, the high water table average rainfall treatments, along with all high water table treatments, had the highest indices, meaning they were wetter and greener than samples with low water table treatments. And I also wanted to point out the difference between hummocks and hollows here. As you can see that there are much larger differences between the indices of each hollow treatment, while the treatment of each hummock have much more similar indices that often even overlap. So again, that's just showing us that hummocks reacted much less dramatically to water stress than hollows did. And then this figure is comparing the spectral indices used to our methane fluxes and net ecosystem exchange. And you can see that the greenness indices, so CI and NDVI, were strongly negatively correlated with NEE. So as the moss was greener and the indices were higher, NEE became more negative, meaning CO2 was being taken in. And you can also see that our water content indices were positively correlated with methane emissions. So as the moss was wetter and the indices higher, more methane was emitted, which again makes sense when you're considering methanogenesis. And I did want to note that boxes K and L seem to display negative correlations between wetness indices and NEE. However, after performing some linear mixed effects modeling, we found that the relationship was based on the strong correlation between wetness and greenness indices, and wetness indices alone didn't have a significant relationship with NEE. And this figure is really the one telling us that not only can spectral indices detect peatland surface moisture and changes in water table depth, but they also relate to CO2 uptake and methane exchange. So this means that spectral indices can reflect the effects of water stress on gas cycling in peatlands, giving us a really valuable tool for understanding, tracking, and modeling the carbon sink status of peatlands in a warming climate. And so in conclusion, we found that spectral indicators of vegetation greenness are most closely related to CO2 emissions, while indicators of water content are most closely related to methane emissions. And we also found that overall, hummocks were much more resistant to water stress than hollows. And finally, we found support for the use of remote sensing techniques to detect water stress and changing carbon dioxide and methane cycling, which could potentially be used in large-scale systems that would otherwise be difficult to access. And tracking these changes are crucial as we enter a period of such uncertainty surrounding the status of these critical ecosystems. So thank you.
Great, thank you, Ali. Finished exactly on time, although we certainly, if someone has a quick question, we can certainly take that now. And again, if people have, if anyone has questions for Ali, then feel free to put it on the chat or quickly ask. I just had a quick clarification question, Ali, and I think you might have mentioned this at the beginning. So for your study site, that hummock hollow classification, like how did you exactly classify? I mean, like looking at the microtopography, or is it like a more subjective based on like first-hand knowledge of the area, study area?
Yeah, that's a good question. So the Forest Service has actually done projects at that same study area before, so they had a pretty good idea of kind of what we were looking for, but it was, I wish I had pictures to pull up, but it was very obvious, like hummocks grow in these like much denser, rounder, like almost little hills, like mini hills, while the hollows are just pretty much in puddles. And then, so we would identify sample sites that way. And then later in the study, we went and did species ID just to confirm that they really were hummock or hollow species. Yeah.
Okay, great. Thank you for that.
Yeah.
And please keep on monitoring the chat, since questions typically can sometimes take a few minutes to pop up. So thank you again. And then if you can stop sharing a screen, we can move to the next speaker, which is Toby Oke from Wildlife Conservation Society Canada and the University of Saskatchewan. Toby, if you could kindly share your screen.
Sure.
We can see your screen. We are not waiting for your presentation to work.
Okay. How about now?
Yes.
Yes.
Okay. All right.
And you can go full screen.
Okay.
Yeah, perfect.
Right. Well, so this talk was derived from a concept paper that we published in last year. The idea is to sort of look at how vegetation is going to shift in sphagnum peatlands and how that will feed back into carbon sequestration. We look at this in the context of nitrogen deposition, warming and drawing down of the water table. We essentially focused on this imagined hypothesis, which is that warming-driven decline in water table coupled with nitrogen deposition is going to lead to relapse in nutrient constraint on vascular plant growth and colonization. And this, in essence, will lead to curtailing of light from sphagnum, causing its decline overall. And this will also lead to increase in microbial respiration and so on. All of this is expected to feed back into carbon sequestration. But there's also an established literature about the facilitative interaction between sphagnum and vascular plant, especially in the area of in preventing photoinhibition, reducing evaporation from sphagnum surface. So how do we reconcile these two perspectives? Like I said, this came from a concept paper, so we did an extensive literature review. So I'm quickly going to summarize what we found here. In terms of nitrogen deposition, we found positive, negative, even neutral for sphagnum. But the effect was always positive for vascular plants. But there was, in most studies, they found that initial stage of nitrogen addition into the system, there was increase in sphagnum productivity, which is kind of consistent with the notion that there's a threshold of nitrogen deposition that's beneficial to sphagnum. Then above that, then it starts to decline. And with regards to warming, it seems like peatlands vegetation is generally resistant to temperature perturbation. In cases where you found increase in effect of warming on sphagnum growth was when they have increased summer temperature by 4 to 8 degrees Celsius, which will beg the question as to whether they are inducing physiological responses in sphagnum that could have occurred in the absence of vascular plants. That is, effect that the response may have nothing to do with vascular plant itself. Now, moisture appears to be the most important factor driving vegetation in this system, and there's inverse relationship between how sphagnum and vascular plant responds to water table drawdown. In the case of sphagnum, with increase in water table, sphagnum tends to expand its vegetation compared to vascular plant. When the water table is low, sphagnum vascular plant tends to have increase in expand their colonization. Now, what's really surprising about all of these studies is that none of them actually quantified light, and light is the main mechanism that they think is going to sort of influence the shift of this influence shift in vegetation one way or the other. And so it's one of those things that we addressed with this paper. The way we did this was we selected 2 sphagnum peatlands with contrasting hydrology. One was fed by the surface water, and the other was more inland. There's no barrier to dispersal, species dispersal between those species. They are within the same climate zone. So we quantify vegetation height as the height of the dominant vascular plant species. As you can see here, with regards to vegetation, in dry peatlands, we really have the vegetation height is really, really low, less than 10 centimeter. But when you compare that to wet environment, it's really high, above 4 centimeter. Again, we also quantify light ratio, where we quantify photosynthetic active radiation above the canopy and below the canopy, and we look at and we express that as a ratio. So high light ratio means that more light was admitted into stratum surface compared to, and low light ratio will mean less light was admitted. As you can see here, light ratio is essentially mirrored the vegetation height, and more light was admitted in the dry environment. Well, the take home here is that light is not necessarily limiting in this environment. And if you think about it, the kind of species that are found in this environment, they are more of Graminoids and Ericaceae family that are really low growing. There are low growing shrubs and the kind of species that can form deep shade in this system, they have high resource requirement. And because sphagnum peatlands are generally nutrient poor, those kind of species cannot establish in this environment. And peatlands also tends to have controls on vegetation change, especially in the area of nutrient immobilization. As we go from the hollow species domination in the relatively hollow peatlands to the more developed peatlands where we have this microtopographic structure, humic hollow structure, we have this vascular peat deposits which prevent nutrient movement through the peat columns. Well, so and with that, the system becomes more acidic, and there's the issue of internal energy balance, where the peat soils become a little bit cold for root establishment. Well, so in essence, as we go from hollow species domination to more humic species domination, we have this inverse relationship with carbon sequestration and sphagnum diversity. That is, as peat accumulation increases, we have decline in sphagnum diversity. But in what way might this system shift as a result of global change? Also, we came up with a conceptual model for potential shift in vegetation and carbon sequestration. Well, so depending on, so depending on where you are, according to global change, you might experience a decline in precipitation or increase in precipitation. At least about 20% increase in precipitation has been projected for some places. So if low precipitation is the case and nitrogen deposition remains normal, well, we expect low water table. We're going to see low water table, and the control of the sphagnum peatland control remain intact. I'm talking about nitrogen, nutrient immobilization, acidity, and internal energy balance, for instance, will remain intact, and sphagnum peatland continue to be the carbon sink. But if the low water table interact with increase in nitrogen deposition, we are likely to see more of a relaxed control, simply because you can see vascular plant expansion and increase in microbial respiration, and the system gradually turn into the carbon source. On the flip side, if the increase in water table, if the increase in precipitation is the case, we'll have increase in water table. And then if the nitrogen deposition is normal, this system remain intact because vascular plant cannot really establish in those environment. But if that interacts with increase in nitrogen deposition, the system still remain intact, but we are going to likely see reduce in carbon sequestration due to microbial respiration that's going to increase as a result of increase in nitrogen input into the system. Well, the take home here is that, well, to sufficiently predict how vegetation might shift in this system requires understanding of regional climate projection, where these systems are found, and having knowledge of plant function, form, and ecophysiology also applies here. And with that, that's it from me. Thank you.
Thank you, Dovey. That's great presentation. Really, it's kind of actually really great to see the interaction between carbon, nitrogen, and environmental variables there. Sure. Questions for Dovey? Any questions? Okay, so Toby, I mean, again, please keep on monitoring the chat as questions may pop up there.
Okay.
And I guess one quick question from my end would be just trying to understand that you have presented this based on looking at a very small local domain, but then how applicable do you think your results, like can you scale up to other regions or other sites within the Panarctic domain?
Well, like I said, this is a concept paper, so we did extensive literature review. This is not just, we didn't base this on the study that we did in the field, we based on the literature review itself themselves. So it's widely applicable. We just summarized literature and we just back that up with what we did at local scale in Southern Ontario. Well, so it's totally applicable. Yeah.
Okay, great. Yeah, thank you for that clarification.
No problem.
And yeah, I don't see any questions immediately. Yeah, sorry, there is a comment from Jackie Schumann, Toby, that was really interesting. It would be great to link this into a model, which, yeah, I completely agree, to kind of try and get this parameterizations.
Yes, that's the plan. And we hope we'll get people, folks to come together to sort of look at that from those perspectives.
Great. Well, so we are just about on time for our break.
Our site, as we mentioned, was present in the interior of Alaska, of which there has been an increase in fire return intervals, or rather, sorry, a decrease in the fire return interval over the last approximately since the 1950s, pretty considerably declined. And as a consequence, we're seeing areas that historically burned between 80 and 150 year intervals on average. Some areas are now burning 30 to 50 years, or even less than 30 years in some cases. And as a consequence, this is really changing the presentation of vegetation in the landscape. It's an area that was predominantly black spruce covered in the past, and now is seeing more representation of angiosperms. And this is likely facilitated by changes in the soil and near ground environment. As a consequence, you're seeing mixed woods develop and deciduous areas develop. And these are areas that change the microclimate and the understory and potentially understory growth. And as a consequence, may inhibit the redevelopment of the soil organic layer, which undoubtedly many of you are aware of. This is a thick mat of organic material that tends to build up in these spruce forests over time and is important to insulating the soil in the summertime, minimizing temperature fluctuations at depth, and in many cases promoting development and maintenance of permafrost. However, when that layer is gone, it's likely that the soils will warm in these areas, and warming soils tend to increase rates of decomposition. And so our project focused on looking at how this increase in soil temperature may have influenced the decomposition rates in these multiple burns. And so we focus on areas that have burned 1, 2, or 3 times in the past 60 years. And the most recent burn all had been roughly 2004, 2005. So all had burned most recently around the same time. We had nested subplots within plots, and these plots were distributed across relatively evenly across these 3 burn sequences. And at these plots, at these subplots, we would place a chamber to measure carbon flux. Now, half the plots were trenched to sever roots, and we also removed any vascular vegetation on the surface. The other half were left intact. And then we also took 2 measurements at any given time. We would measure initially with everything covered, and that would allow us to measure just the respiration of the soil and any plants underneath. And we also took a measurement with the cover off, which would allow for the photosynthesis to occur and would measure the net exchange of carbon from that segment of soil and plant. The difference between the two, of course, would then be gross primary production. Now, this talk is going to focus specifically on trench plots that were covered. So we're going to look just at the changes in heterotrophic respiration in these environments. So we also assessed the depth of the soil organic layer. We did some much smaller scale vegetation surveys than Kate Hayes had done, but we also observed a very rapid decline in the soil organic depth with burning, particularly by the time you got to 3-time burns, it was essentially non-existent, a thin little smear on the surface. Whereas because we focused on areas that were more covered with overstory, we saw an actual increase in deciduous overstory cover with burn frequency, especially by the time you got to 3-time burns. And we have there are 2 sites here. Our Dalton site is roughly equivalent to our upland site, and our Stee site is roughly equivalent to the lowland site that Kate was looking at. We also looked at root depth, or rather we looked at biomass of roots at depth in the mineral soil, and we found that there was generally a substantial increase with reburning, and that it's hard to see on this particular figure, and the GV VASC in this figure denotes the biomass of ground level understory vegetation, so vegetation under 30 centimeters, the stuff that would have ended up being in the chambers where we didn't trench. And we also saw a significant increase in the one-time burn, or higher amounts in the one-time burn from the controls, the unburned areas, but a rapid decline, particularly in the 3-time burns, where it was essentially non-existent. And this gives you a similar visualization of what those burn progressions look like. So on the left, you have your mature unburned forest, roughly about 100 to 120 years old. After 15 years after a burn, you can see there's a lot of standing dead trees and actually a large amount of a low understory reappearing. 2-time burns, it's depending on the site, it's a little more complicated, but we start seeing a lot more deciduous shrubs and trees come into play. And by the 3-time burns, we saw a lot more homogeneous deciduous tree cover, including birch and aspen. Looking at the differences in soil temperature, it's a little hard to read on this, but you can see that the 3-time burn by far had the deepest soil temperatures over the course of our measurements, which was during the growing season, and followed by the 2-time burns, and then the 1-time burn and the mature forest were fairly similar at the 10-centimeter depth. Our measured fluxes, if you look on the left, we had measured fluxes from one site over a period of 2 years, and on the Stee site only over a period of 1 year. And you see this progression of increasing flux. So the green lines are the total respiration, and the orange lines are heterotrophic respiration. So we looked at this, I took this, and we were unable to measure data that were at the shoulder season. So we were not able to predict what flux might have been in that period. Using some values from the literature, including soil temperatures, we developed a mixed model that allowed us to estimate flux at hourly intervals. And that's the figure you see on the right. And it matches up with what we observed in the measured fluxes quite well. And this was based on using soil and air temperature as predictors. And that accounted for approximately 74% of this variance in a pseudo R square. Now, if we ignore the sites and we bring all the data together and we look at the annual flux patterns, if you focus on the orange lines here in the first panel where it says flux, you can see a large decline in flux in the 1-time burns from the controls, and then a distinct increase over the burn frequency. So by the 2-time, you've increased by about 28%, and by the 3-time burn, we've increased by as much as 80% over a 1-time burn. The bottom shows differences in GPP, and the panel on the right shows what I'm loosely referring to as net ecosystem exchange, just from the 30 centimeters and below. This does not include the overstory. So the general point is that you're seeing that there's an increase in respiration from heterotrophic activity, and that is pushing, at least at a ground level, the exchange of CO2 from the surface of the earth. Now, our model did a good job of predicting that. However, that was only when we set having a separate intercept for each treatment. If you look at each of the fixed effect values, if we adjusted the model by just say predicting the same temperatures at the 1-time burn, it only actually accounted for about 30% of the difference between, or less than 30% of the difference between the 2- and 3-time burns in terms of CO2 flux from heterotrophic respiration than the 1-time burns. So that means there was a lot to this model or a lot to this pattern of flux that's not being accounted for simply by surface temperature alone. So if we took a look at relationships with other covariates like soil organic layer depth, the amount of deciduous tree basal area, and the root biomass in the mineral soil, first in the first row, we see the relationship to soil temperature, and predictably, less soil organic layer depth led to higher soil temperatures. We saw greater soil temperatures where you had actually more deciduous overstory. and you had greater soil temperatures where you have more root mass. Now, clearly that's not going to be a causal relationship, at least the latter one. But if you look at the flux of CO2 from the surface, or again, we're going to focus on the orange lines here because that represents our heterotrophic respiration. We see that there's predictive ability from some of these, particularly things like rooting depth, that predicted much higher flux in areas where you had much higher root biomass. And it's not present here, but two other things of note is that we have some limited measurements of soil temperatures at 50 centimeter depth. And it was both higher at the one-time burn than it was at the mature by about 2 to 3 degrees, but it was actually substantially higher in 2 and 3 time burns, about 4 to 9 degrees Celsius higher than it was in mature forest, and about 3 to 4 degrees higher than it was in the one-time burn at 50 centimeter depth, which suggests that more soil organic carbon is now available for decomposition in the 2 and 3 time burn areas relative to, say, the one-time burn or mature forest. And take into account that we have this increase in heterotrophic respiration despite this loss of this thick organic horizontal surface that would otherwise normally be contributing in part to that. So the overall, story here is that reburning seems to be increasing decomposition, or potentially increasing decomposition, which is leading to this increase in heterotrophic flux. From our research, it's currently unclear if that's going to be offset by an increase in overstory biomass in the deciduous areas. Clearly from Kate's research, a lot of these areas are not putting on more deciduous biomass. So in those areas that may not account for that, but we did not specifically target these areas that did not have a large amount of overstory. So as a consequence, they may not be turning over that soil organic carbon as much. I'd like to thank a lot of people.
Hi, Jason. Please.
We're good?
Yeah, I think, Jason, if you could kindly wrap up,
okay.
That would be great.
Yeah, I'll end it there. Thank you.
Thanks, Jason. Sorry, didn't mean to necessarily cut off on your acknowledgements, but just being mindful of the time and the upcoming speakers. So again, if there are questions for Jason, if you could kindly put them in the chat, and then Jason, if you can monitor the chat as the questions pop up, you can address them there. Thank you. So our next speaker is going to be Jonathan Wang from University of California, Irvine. And John, if you could kindly share your screen.
See it?
Yes, you see it. Perfect.
Okay, great. I'll just start.
Thanks, John.
Hey, everyone. Thanks for having me. I'm John, coming from UCI, but also representing some work that I've been doing as part of my dissertation at Boston University. And so what I'm here to talk about is this paper we recently published where we looked at using remote sensing data sets to look at above ground biomass across the above domain. And so we're gonna change gears a little bit, look at some larger sample. This will be interesting just in terms of seeing how these biomass and carbon dynamics really scale up and how they are reflected or altered by these disturbance regimes. So some of the motivation for this work is really focused on this notion of northern greening trends, right? And so if you zoom out and look at the whole Pan-Arctic, Pan-Boreal zone, we can see that based off of trends of NDVI and these remote sensing based indices of greenness, there tends to be a general increase in this value, that's NDVI, which suggests an increase in plant growth, right? And so there's some mystery around why this might be happening, but people are supposing so far that it has to do with the warming, climate warming, or CO2 fertilization, leading to an increase in vegetative growth in the high northern latitudes. And so we're here to try and investigate this with a little bit more of a direct measure of above ground biomass or carbon, let's say. And so the reason we're very interested in knowing a little bit more about the carbon dynamics here is that we know that in the northern ecosystems, there has been a pretty significant regime of disturbance. So wildfire and harvest, I'm showing on the left for Canada's managed forests. And on the right, there's evidence of extensive land cover change, right? Losses of evergreen forests in the red. So that's some of those black spruces burning up that people have been talking about in the session, as well as some evidence of increased deciduous forest cover in the further southern parts of this this domain, which is the NASA above domain, right? So this is where most of my work will be focused on. And so what we're going to try to do is we're going to try and measure forest biomass, above ground biomass. And so the way this is typically done in the field is that we will go into the, to a plot, measure tree structure, diameter, height, and convert that to biomass using allometric equations. But the issue here is that it's really hard to get, let's say, a regional scale idea of biomass dynamics using this method, because it's pretty hard to get out to these field sites regularly and across a large enough area to really understand what's going on. And so lately, in the last, I don't know, let's say 10 years or so, there's been a lot of advances in our ability to use remote sensing to scale up these allometric equations to sort of the regional scale. So here, just, this is pretty far afield for a conference focused on boreal forests, but just as an example, this technique has been used to measure tropical forest carbon dynamics and how they've changed as a result of deforestation, for example, in the Amazon. And so the research questions I'm focused on here are really, there's two. So the first I'm really trying to understand is how have these disturbances, I'm talking about fires and harvests, how have they reshaped boreal forest biomass dynamics in recent decades? And how well do current Earth system models capture these dynamics? And then what can we learn about how these models work by bringing to bear these large, dense remote sensing data sets? And so just a quick overview of the methodology. The way we do this is that we use spaceborne LiDAR from the GLASS instrument flying on the ISAT satellite, and combine that with allometric equations, right? So at each shot, we try and get an estimate of above ground biomass and come up with this sort of inventory of above ground biomass across the whole world. We're just going to focus on the above domain though. And we combine that with, I'm calling it CCDC smooth Landsat time series, but you just need to worry about the fact that it's a high resolution 30-meter satellite measuring surface reflectance at across many decades, 1984 through 2014 in the case of this study. And so we combine this biomass data set with this Landsat observations to create time series maps with machine learning of above ground biomass. There's a little bit of statistics that we throw out there at the end to improve our estimates just a little bit. We won't worry about that too much. But the end result is that we have this annual biomass time series, above ground biomass time series at 30-meter resolution for about 31 years, 1984 through 2014. Just a quick note on above ground biomass and LiDAR. So this, you might not be familiar, we're using GLASS, flown on ISAT from 2003 through 2009. I'm showing here on the bottom panel is all the individual shots, right? So it's not mapping, it's a sampling satellite. And so at each of these shots, we get these sort of LiDAR plots that give us biomass. And we try to extend that, or maybe extrapolate that through space and time using remote sensing from Landsat, as well as machine learning. And I won't sweat the details, but basically we get a pretty good fit, about 0.8 R squared, 20 megagrams per hectare RMSE. So we feel pretty good about our ability to map at least what GLASS thinks the biomass looks like using Landsat inputs. And so what we're finding is, disturbances such as harvest in this example are occurring at short spatial scales, maybe on the order of hundreds of meters. And so this high resolution data is really important for delineating, just when and where changes in biomass occur as a result of some of these disturbances, right? So here in this example in British Columbia, we can see these dark green areas where there's dense forests getting replaced by these, patches of low biomass areas as a result of harvests throughout this area. And if you pay careful attention, you can see regrowth, the accumulation of biomass at previously harvested areas slowly over time. And what we end up having is this sort of complex mosaic Some areas have biomass losses in the browns, and some areas have biomass gains in the greens, occurring both at this sort of landscape scale as well as sort of the regional scale. And so this is the whole domain that we've mapped. On the left, I'm just showing the average biomass density across the above core domain, excluding the tundra. And on the right, I'm showing the net change in biomass as a result of growth or these disturbances removing the biomass. And so that's using similar color scales as before, with the browns being losses of biomass and the blues on the right being increases. And what we notice at this scale is that the sort of patches of biomass losses tend to spatially align if you squint at some of these instances of fires. And so we're looking at fires, and the red and purples indicating the perimeters of burned areas from the Alaskan fire database and the Canadian fire database sort of combined. You can also see, you know, with the fires burned earlier than our record, we tend to see increases in biomass. So for example, some of these purple areas have these sharp, deep green areas. At the same time, we have these harvests that come from the Canadian Forest Service Landsat product, and they're showing in the blue in the southern portions domain a lot of spatial sort of complexity there as well. If we sum up across the whole domain, we see you know, roughly an increase in above ground biomass, but there's a lot of this sort of temporal complexity you know, across the 31 years. Over the first 20 years or so, there's been this strong increase in above ground biomass that gets rapidly reversed in this big fire year in 2003, 2004 in Alaska. And then it sort of comes back later in the later part of the domain, or sort of the period. We have a net gain of about 400 teragrams of biomass over this whole 31-year time span. We separate out the fire, just the fire affected areas, we see that the losses outpace the recoveries, so the post-fire recoveries, and the net losses are about 147 teragrams of biomass. With the harvest,
hey, John?
Yes.
Hey, you're at the 9-minute mark.
Okay.
You have about 2 minutes to go. Yeah.
Okay, I'll zoom through the rest. So, so basically, the long story short, 30% of the above ground biomass accumulation that would have occurred, it's been suppressed by recent disturbance, fires, and harvests. And so here's a very complex plot, which I don't have the time to explain, but the basic message here, just to cut to the chase, is that Earth system models overestimate this above ground biomass gain by a factor of 3. And we think that this is because that they poorly represent fires, sort of fire dynamics throughout the domain. And if you're interested in more details, I'd be happy to talk to you more about it. So basically, above ground biomass has increased and above about 400 teragrams. Fires have canceled recent growth, and there's a lot of mismatch between this biomass growth and stocks simulated by Earth system models. Thanks. Sorry for going over.
Thanks, Sean. That was great presentation. Yeah, so are there any questions for John? John, I actually had one. If you could just go back a couple of slides, there was a lingering statement on how fires are poorly represented in Earth system models. So what is it like, or what are the processes that are not being captured, or what parameters are poorly represented? Is it just like the drivers of fire, or is it both like the trends and how those drivers will change and how that will impact the trends, or is it like a combination of everything?
So we didn't get into the processes very much because I'm not really an expert at Earth system models, but we found that there weren't that many models that predicted or simulated fire areas to begin with. So it was hard for them to capture these changes. And the ones that do predict fire areas in this, well, I guess in general, because they're all global, you can see on this panel on the left, you know, this black line is what we saw in the fire databases in terms of the total area burned per year. And the majority of them don't capture the magnitude at all. They're off by, I don't know, like a factor of 5 or 6 or something. And the one that does happen to be in sort of the same magnitude, order of magnitude, doesn't capture the trend. There's no, you know, there's this increasing incidence of big fire years, and CSM2 is just flat. stationarity across this. So we think we'll leave it up to the Earth system modelers to figure out the processes, but we just noticed that the trends are not being captured here in the burn area.
Okay, fair enough. Okay, and any questions for John, please put them in the chat. And then John, if you could also just keep on monitoring as questions pop up. Thanks again. Thank you. So our next speaker is going to be Manuel Helbig from Dalhousie University. And Manuel, if you could share your screen.
Okay, can you see
and
it?
I guess we'll hear some, yes, we can. And we'll hear some insights about how our climate models are doing.
Okay. Yeah, thanks. I'm Manuel. I'm an assistant professor at Dalhousie University. So I'm coming all over from Canada. And my title was a bit different. So my title was looking at temperature sensitivity of net ecosystem exchange in boreal forests and peatlands. But today's talk will really focus more on the peatland section of this talk. And I would like to start with just like the reason why, or one of the reasons why we talk a lot about peatlands in the boreal biome is because they really are long-term net CO2 sinks. So every year they take up a little bit of carbon and put it down into the peat column and accumulate it there. But what is really important is that there's a seasonality to that flux. So basically the net sink is really just accumulating all the daily fluxes, and the winter has a specific signal. So this is an example from the Hudson Bay lowlands for two different peatlands where you have winter respiration, no vegetation productivity. So you lose CO2, and then with a snow melt, you suddenly start taking up more CO2, but in some cases you also increase respiration, so that outcompetes the productivity. And then the peak growing season is usually when you take most carbon up. Yes?
Manuel, we're still seeing your title slide. So in case you intend to move to the next slide, then it hasn't moved forward.
Sharing is paused. Resume share.
We are seeing seeing your PDF, actually, the Acrobat Reader app software.
Let me just try to just share this screen here and not. Do you see it now?
Okay.
But
okay.
now
yes.
Perhaps put this down and then. Okay. So
we now see slide 1. Yes, great. Thank you.
Perfect. So we have the peak growing season where we take up most of the CO2, and then towards the fall again we lose more CO2, and then the overall result is that there is a net uptake. But so what we ask in this study is what happens if we change temperature, and we know the temperature is not uniformly warming across all the seasons. So combining the temperature sensitivity for each season with the actual observed change rates in warming. So how we can do this? So we use eddy covariance data from multiple sites. We use at least 5 years of data to get interannual variability. And then we work with monthly data. And then if you look, for example, at the time series, this is just visualized in colors. You could look into individual month. You look into the net CO2 exchange, and on the x-axis here, the air temperature. And then you just, with a simple empirical relationship, you look into, does actually net CO2 uptake increase or decrease with warming? So here, that would be a increased net CO2 uptake because it's a negative relationship. That would be a positive relationship, which would be a decrease in the net CO2 uptake. So we apply this approach to a tower network, kind of a synthesis project, gathering peatland data from eddy covariance sites from 19 sites, with in total over 2,000 site months. As I mentioned before, minimum is 5 years. You see, we have quite a good coverage for the boreal biome in North America. So Europe is very well covered in terms of the peatlands there, and then unfortunately we have this large gap in Siberia and in general in Russia. And we cover quite a range of mean annual air temperatures and precipitation. So now really, when we look into, take a step back and also think about, are there any experimental results that we could look into? So there is a spruce experiment in northern Minnesota. It's a peatland that has been warmed with different levels. But really important is that also the warming is uniform. So basically each month has a +2 or +4 or +8. So there's no seasonality in the warming. And if we look here at the NEE changes in relation to the temperature anomaly, we see the red line. It's really a strong relationship. There's a very clear decrease in net CO2 uptake with warming. But then if you're at the eddy covariance side, you look at interannual variability, you actually have seasonally varying warming and cooling and so on. So when you then look at the anomalies across all these sites, you see that we actually can only explain less than 10% of the variation in the interannual variability. And that might be partly because of the differences in warming patterns across different seasons. So now when we use this big synthesis data set, and basically we do a linear mixed effect model, we take sites as random effects. And so we have the fixed effects will be the relationship between mean air temperature and net CO2 uptake. And then we plot this against the mean air, we bin the data according to mean air temperature for different months for these sites. Then what we see is when we here on the top, you see the first half of the year, you see that there's a small positive relationship, means less net CO2 uptake with warming. And then you see pretty much about 0, you start with that response. There's more net CO2 uptake with warming. And then you hit like a peak. And then towards the really warm temperatures, you go again towards almost like a non-sensitive or non-sensitivity to temperature. And then if you look at the same pattern in the second half of the year, so that would be late growing season, fall and winter, early winter, you see a fairly similar pattern. But if you zoom into the late growing season and compare it to the same temperature in the early growing season, you see that in the fall or late growing season, we really have no change in CO2 uptake with warming, whereas we have enhanced CO2 uptake with warming early in the growing season. So that tells us there is a distinct seasonality in this temperature response of net ecosystem exchange. And that really depends also on the season. So there is kind of a hysteresis across the year. So now the next step is we look into, because we know there are direct temperature effects, for example, in affecting photosynthesis or respiration, but there are also indirect effects. So it can, for example, it can affect the leaf area index or in general the vegetation productivity, and we can use enhanced vegetation index to look at that. And also we know peatlands are very sensitive to water table depth, and there is a link between water table and temperature. Usually with higher temperatures, you have more evapotranspiration. So now if we plot basically what we see here on the left side, we do the same analysis with enhanced vegetation index and water table depth. Again, on the top here, the first half of the year, on the bottom, the second half of the year, then we see that there's a strong correlation here for enhanced vegetation index, but only for temperatures. So we use a threshold analysis only for temperatures below about 11 to 13 degrees. So that means early and late in the growing season, it seems like it's really the temperature sensitivity is really linked to the sensitivity to EVI in this case. And then if we look, there's a significant relationship between the temperature sensitivity of NEE and the water table sensitivity of NEE. It explains less, but it's significant, but it's only for the warmer temperatures. That means so in the peak growing season, really it seems that the air temperature effect on water table is contributing quite a bit to this pattern. So now we take this one step further to ask the question, what's the impact of seasonally varying warming on annual NEE in peatlands? So what we do is we take these sensitivities for each month across all these sites. So that's our best estimate for temperature sensitivity for NEE across all these peatland sites. So we see that seasonal hysteresis here. So with increasing uptake, and then loss in the early growing season and a loss in the late growing season. We couple that to recent warming trends, so from 1951 to 1970 and 2001 to 2020. We take the climate research unit temperatures, and then you can see clearly here for January, March, April, June, July, September, October, December, you see very distinct patterns in both spatially but also seasonal changes in warming. We know that winter warming is the strongest, but then you see also strong spring warming, for example, in Siberia. So now if we couple these two, so we know the sensitivity, we assume that they're in all the peatlands the same, and we do this for each month and sum up what the change in NEE would be, then we get these maps here where you see the estimated change in NEE due to temperature change. And then you see here quite like really this reflection that there appears to be a strong or an estimated strong uptake in Siberia, whereas in Europe we see a decrease, and then also a diverging pattern in North America. If we assume a seasonally uniform warming, so every month has basically the average annual warming rate, then there is really no difference. So there will be just a general decrease. So what you see? Yeah, I'm almost. That's the last slide. So if you look at the warming in the early growing season, April to June, and you look on the left side, this is the seasonal varying warming. You really see that the strongest uptake we can expect in the areas that have early growing season warming, whereas when there is almost no warming, we expect probably losses in net CO2 uptake. So really the regions where early growing season warming dominates increase the net CO2 uptake. Thanks.
Thanks, Manuel. And any questions, quick questions for Manuel? I think there is one comment on the chat. Not all ESMs show GPP increase in historical experiment everywhere. I'm not sure if that's in relation to this one. See GPP trends for Southeast Asia, or if it is in response to the previous speaker, it's not clear.
Okay.
But yeah, one question is like, are you also exploring the role of nutrient limitations, so like nitrogen deposition?
Unfortunately not. It came up when we thought about how we can analyze the synthesis data set, and yeah, I think perhaps we would need a better spatial coverage or better estimates of nitrogen deposition. But it's true that probably a lot of the sites in Europe will experience much higher nitrogen deposition than some pristine sites here in North America. So something to look into.
Yeah. Okay. Great. Thank you so much, Manuel. And then moving on to our final speaker for this session, it's going to be Peng Zhao from Swedish University of Agricultural Sciences. And I believe Peng will be able to shed some light on the role of nitrogen, on how it alters responses of ecosystem, carbon fluxes to environmental drivers. Peng, we can see your screen, and if you can, yes, perfect. Thank you. We can't hear you though, Peng. Peng, you are still muted. Helen, can you see if you can unmute Peng?
Working on it.
Okay, thank you.
Where are you? Oh. Bang. Okay, as to unmute. As to unmute. Okay. All right.
Peng, can you speak now? Maybe we can hear you.
We don't see your screen anymore. Bang. Okay. Can you talk to make sure? Okay. Okay, we hear you.
Yes, we hear you now. Thank you, Peng. Thanks, Alan.
Yep.
Yeah, Peng, if you can go full screen, that will help. Peng, we don't see your screen anymore now.
So, Peng, there is two ways we can go at it. Whether you forget about presenter mode and you present your slide this way, or I can share your slides and you tell me when to switch slides.
Hello, can you hear me?
Yes.
Yes, we can hear you. Great.
Now we need you to share your screen.
Okay, sorry everyone, sorry to the audience for the technical difficulties. We hope to resolve it soon.
Okay.
Hello, can you hear me?
Yes.
Yes, we can hear you.
Oh, okay, sorry.
That's fine. Yeah, if you could just go full screen and start. Great,
Okay.
Thank you.
And can you see my screen?
Yes,
yes.
we can.
Okay. Today I'm going to talk about our results of the long-term nitrogen addition on the forest ecosystem scale carbon balance in a boreal pine forest based on eddy currents measurements. As we know that boreal forests are commonly limited by nitrogen, and nitrogen fertilization has been applied in some parts of the boreal region. And in addition, the elevated nitrogen input has also resulted from the increasing atmosphere deposition during the past decades. And all this increased nitrogen input could enhance the boreal forest carbon sequestration. However, most of the previous studies focused on the increase of above ground tree biomass and soil carbon stocks based on mainly on plot scale experiments, but the effects on the ecosystem scale carbon balance are lacking. That's our study based on eddy currents at both the fertilizer and the control stand, focused on the long-term ecosystem scale fertilization on the forest ecosystem scale carbon balance. And our experiment site is located in a pine forest in northern Sweden. We have two stands that control and fertilize. The fertilization started in 2006 and was annually applied through helicopter. And meanwhile, we have also the eddy currents measurements of the above canopy CO2 fluxes starting from 2006. And we selected two study periods. First is in 2006 and 2007, that is the first year of fertilization, and the other 5 years after 1 decade of fertilization. And based on these measurements, we focus on the long-term fertilization effects on the forest ecosystem scale carbon balance. Here is data processing. In this presentation, we will focus on the CO2 fluxes, including NEP, GPP, and ecosystem respiration. First, we compare the meteorological factors from 2006 to 19, we can see that the first study year, 2006, was a normal year compared to other years, where the 2018 was a drought year with less precipitation but higher temperature. Okay, some results. This figure represents the annual CO2 fluxes at the fertilized and the control stand in 2006 and from 15 to 19, and also the response ratio between two stands. First, we can see that in 2006, the control stand had both higher NEP, higher GPP, but similar respiration compared to the fertilized stand. And after 1 decade of fertilization, the fertilized stand has both higher NEP, GPP, and respiration than the control stand. And if you look at the response ratio, you can see that all the carbon fluxes in 2015 to 19 didn't further increase because the response ratio was relatively stable in these years. And then we look at the monthly flux difference of NEP, GPP, and respiration between two stands in 2006. Here is represented by the red bars, and 2015 to 19 blue bars. We can see that in 2006, the control stand has both high NEP and GPP during most of the growing seasons, and the maximum difference occurring in June. And after 1 decade of natural addition, the fertilized stand had higher NEP and GPP during the growing seasons, with the maximum difference shifted from June in 2006 to August in 2015 and 19. And we also found that long-term natural addition also changed the fertilized stand to become a larger respiration source during the whole year, where in 2006, the respiration had no difference between two stands. And then we analyzed the relationship between total NEP difference in winter against summer. Here, the positive numbers axis represent numbers at the y-axis represent higher winter loss. Thus, we found that long-term natural fertilization not only enhanced summer carbon uptake, but also increased winter loss, indicating a composition between the growing season uptake and non-growing season emissions. And this cross-seasonal feedback further resulted in a smaller difference in annual NEP from 2005 to 15 to 19, compared to the bigger difference in GPP and respiration. And then we analyzed the daily flux difference against several environmental factors, including radiation, air temperature, and BPD. Here, the figures from A to C is for NEP difference, and D to F is for GPP, and the upper three figures is for respiration difference. Positive numbers means higher fluxes at the fertilized stand, where negative numbers represent higher fluxes at the control stand. We can see that in 2006, the difference of NEP and GPP increased with both radiation, temperature, and VPD, but leveled out at higher temperature and VPD. However, after 1 decade of but in 2006, the respiration difference has no significant difference. And after 1 decade of fertilization, both NEP, GPP, and respiration were higher at the fertilization. And specifically, we found that the GPP difference increased with radiation, air temperature, and VPD, but leveled out at higher meteorological values. However, the respiration difference in 2015 to 19 continued to increase with these environmental factors. And this different response of GPP difference and respiration difference in 2015 to 19 further resulted in that the NEP difference first increased with respiration at temperature and the VPD, but then declined at higher meteorological values, as we can see in figure A, B, and C. Okay, some summary points. In the first year of fertilization, the control stand has both higher NEP and GPP, but similar respiration than the control stand, than the fertilized stand. And after 1 decade of nitrogen addition, all the carbon fluxes at the fertilized stand exceed that of the control. We also observe that the greater growing season carbon uptake is compensated by increased loss during winter, and long-term nitrogen addition alters the responses of the carbon fluxes to environmental drivers and shifts the carbon balance of the Boreal forest to a new steady state. Thank you.
Are you around, Abhishek?
I don't hear Abhishek, but I can, I can ask some questions in the chat. Thank you for the nice presentation. Thanks, and sorry for the technical difficulties there. So Riza Makapai asks, was our eco studied in bare plots or our soil?
The total ecosystem respiration, including both the CO2 fluxes from the soil and also the above ground trees, total respiration.
Hi, hi Peng. This was actually a question of me, Boris Stupek, but I'm also from Finland working with Riza. But this respiration of the soil, was it trenched or did it include tree roots and understory vegetation?
Sorry?
In soil respiration, because you are saying, yes, it's ecosystem respiration is part of, maybe I show also myself. So the ecosystem respiration is part of all the components, but the soil respiration, was it separated from autotrophic respiration?
In my study, we don't have the soil respiration measurements. Because we have only the eddy covariance measurements. That's the respiration is the total ecosystem respiration.
Yeah, eddy covariance is not ecosystem exchange. So how did you establish these relations with respiration if you didn't have respiration?
First, we have the measurements of the net CO2 exchange, and then the NEE was separated into GPP and ecosystem respiration using the respiration models.
Okay, I see.
Yeah.
So yeah, because there was a different response to these different study sites, but part of it was based on assumption on this separation, I think. Okay, thank you.
Thanks, Boris. Sorry, everyone, my audio is back now. I think there is one more question from Riza on what was the stand age on your study site?
Stand age is around 90 years old now, from about 90 years old. The forest was established in almost 100 years ago, 1920s.
Okay. Great, thank you, Peng. And I think we are slightly over time here, so I apologize for some of the technical difficulties we had at the very end. But again, I think overall this was a great session. So thank you to all the presenters. Thank you to the audience. And also thanks to Helen, Nick, Scott, and everyone for contributing and attending this session. Helen or Nick, any final words before we wrap up?
No, I just wanted to thank you all, and thanks to the presenter. It's no big deal that we went a little over time. It's the last event of the day, so we can wrap up for the day and see you all tomorrow at earlier than today to accommodate our colleagues in Europe and on the East Coast and in Russia and in Asia in general. So see you tomorrow for another great conference day.
Yep, absolutely. Thanks, everyone. Thank you. Bye now.
Bye.
Thanks, everyone.
Thank you. Bye.